Okay, this is a short video to demonstrate gene expression. Here we have the DNA molecule. Hopefully now you're recognising its double helix shape. Two strands, which we'll describe as anti-parallel. This strand in the bottom starts with a phosphate, so we should know that that's going to be the 5 strand, making this the 3 end. And the other side starts with the deoxyribose sugar, so that will be the 3 end, making this the 5 end. The nucleotides are connected between the different strands with hydrogen bonds between complementary bases. The nucleotides in the same strand are joined together with a sugar phosphate chemical bond, and this together forms the sugar phosphate backbone. So we'll just draw the sugar phosphate backbone. The genetic code here is what the cell is going to use to code for a protein. So the first thing that needs to happen is we need to unwind and unzip the DNA. The enzyme that does this is called. So DNA polymerase is going to come along and it's going to unwind and unzip the DNA, and that exposes the bases or the genetic code. What's going to happen next is the RNA polymerase is going to add and join RNA nucleotides. So the RNA nucleotide is similar, except it's got a ribose sugar, and instead of a thymine base, it will have a uracil base. So on the other side, we're going to get, if that's a C, this would be a G. What I'm doing right now with the pen is what the RNA polymerase is going to do. It's taking 3 RNA nucleotides and it's using complementary base pairs to join them against the DNA template. It then joins the nucleotides together to form a strand of RNA. Note this is the 3 end, so this is the 5 end of the mRNA. So what we're doing here is we're joining nucleotides always to the 3 end of the new strand. So when the RNA polymerase has finished, the primary transcript of mRNA detaches from the DNA and the hydrogen bonds can reform and the DNA will join back up. So we'll re-zip and rewind. And that's as done with the DNA. This first molecule that we've made then is called the primary transcript. Now, this is only 6 bases long. In reality, it'll be much, much, much longer than that. So, another way of looking at it, a much larger size then. This box represents many, 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 many bases in this base sequence. So what we need to do next is turn the primary transcript into the mature transcript. To do that, a process called RNA splicing takes place. The primary transcript is made up of different sections. These sections can be called introns and exons. The introns are removed by enzymes. So these introns are cut out. The introns are called introns because they stay in the nucleus. So the introns are non-code. They are removed. When we remove the introns, and you can see this enzyme action here, so let's remove that intron, and that intron. So these are removed and taken away. We're only left with the code. So we call the code sections, all the code sections exons. The final stage of RNA splicing then is to join these exons together, because the mature transcript consists of only exons. Now we could number this exon 1, exon exon 2, exon 3, exon 4, exon 5, exon 6. So this mature transcript has exons 1, 2, 3, 4, 5, and 6. We can actually make different mature transcripts from the same primary transcript depending on which exons are retained. We don't have to keep exon 6. So this would be a shorter mature transcript. Or we don't have to retain exon 1. So again, we've got a third version. We can actually remove exon 3, and as long as we remember to join 2 and 4 together, we have yet another mature transcript. So we can keep making different mature transcripts from the same primary transcript, just depending on which exons are retained. It's important to note the order of the exons never changes. So let's look back at our transcript. We'll say now that we've removed any non-code sections, so all the introns are gone. This is only exons, which makes it the mature transcript of mRNA. So the ribosome is made out of two parts. It's made out of rRNA and it's made out of proteins. So we've covered transcription in nucleus, we've covered RNA splicing. The next and final step is translation, and this happens at the ribosome. Okay, so the mature transcript of mRNA arrives at the ribosome, and the way this works is it's read 3 bases at a time. So we call these 3 bases on the mRNA a codon. So our first codon is CUG and it arrives at the ribosome. What has to happen next is we have to get the correct amino acid. So another molecule of RNA called tRNA, which has the correct amino acid, will come along and it will attach to the mRNA. So the marble represents the amino acid. The tRNA also has 3 exposed bases, and this matches because of complementary base pairing with the codon, so we call this the anticodon. The one that will match with this will be CUG. So if this anticodon was different, it would not be able to bind. This has to be complementary. So G, U pairs with A, G pairs with C. So that tRNA, the anticodon, can bind to the codon of the mRNA using complementary base pairs. That means the correct amino acid has been transferred to the ribosome. The tRNA, once it's left its amino acid, floats away, and the mRNA can move on to its next codon. Now the codon here is AUA, so we're going to match that, the complementary base pairs, to a tRNA with UAU. So different tRNAs will be in the cytoplasm. The one that's complementary to this one is going to come down, join on with the complementary base pairing, and it brings the specific amino acid that comes second, that's coded for by the second codon. Again, this detaches, leaving behind its amino acid, and now we have a second amino acid, and the mRNA keeps moving down. So, we'll do a couple more codons. The ribosome now has 2 amino acids. These amino acids are held together with peptide bonds. So the ribosome's joining a peptide bond between the amino acids. Okay, so our next codon is UAG. So we need a complementary codon that matches with that. So we're going to need one that has an AUC. The tRNA molecule with the matching anticodon will come down with the next correct specific amino acid. Again, that's joined, so the tRNA can leave again. And another peptide bond is formed between the amino acids. So now we've got 3 amino acids. The mRNA moves down to the next codon. This one is CUA. So we're going to need one that has a GAU. The tRNA with the matching anticodon, which means that the complementary base pairing can occur, brings the next correct amino acid. mRNA keeps moving on, the tRNA leaves, and we get our next amino acid. This continues on and on until we have reached the final codon. The final codon doesn't in fact code for an amino acid. The final codon is called a stop codon, and it stops translation. So at that point, no more tRNAs will match. The mRNA returns to the ribosome. And here we have a polypeptide chain of 3 amino acids. So I'm going to dissolve them. So here we have our 4 amino acids. They are joined together with peptide bonds. Because these bonds are called peptides, and there's several of them, we call this chain of amino acids the polypeptide chain. So poly means several, peptide because there's several bonds, so polypeptide chain forms. Don't get that mixed up. The polypeptide is the molecule, the peptide is the type of bond between the amino acids. This isn't yet a protein. The final thing that has to happen is that certain amino acids are attracted to one another and they want to be close together. These are held with weak bonds, such as hydrogen bonds or other molecular interactions, which you don't need to know, so just stick with hydrogen bonds. That's going to cause our chain to fold up into a shape. So if I was to redraw that, this amino acid, because of a hydrogen bond, wants to be close to this amino acid. So now it's folded into that shape. That's key. We're going to use the word fold into the correct 3D shape. And once it's folded into a 3D shape, at that point, we can refer to it as a protein.